And I will disclose right away that I'm certainly no expert on pain. And everything I know about pain has to do with your keynote lecturer yesterday, my colleague Vanya Apkarian, with whom I've worked at Northwestern University before I moved. And we still discuss, we still meet. And what brought us together was really the realization that pain, which Vanya was studying in the model of spared nerve injury, and learning to be afraid actually have much more commonalities than one would think. So we slowly, through our walks around the university day after day after day, decided to test this with some common projects and some common shared work and see whether indeed pain, and especially chronic pain, which doesn't have obvious triggers, ongoing triggers, could be related to the formation of pain memory traces which become reactivated throughout individual's life. So I will have two parts in my talk, which are, the first is about memory, at least my view on the memory. When my views deviate from the established, I will let you know so that you don't take it as a common truth. And then I will have a small selection of data, which in my view were the most fascinating, bringing the two fields together, and conclude with some analogies about how processing of pain may relate to the activity of the episodic memory system. I work on the hippocampus. Here it is. And for many years, we've worked on the mechanisms in the hippocampus in memory formation. We then expanded to the more extended hippocampal circuit, and especially the activation of this cortical area. I don't know, just to mention, although we are all talking how for the memory formation, we need the hippocampus and we need the cortex. And based on that, we initially thought that the hippocampus is extensively connected to the cortex because we know that memories are spread and widely represented in the cortex. And it was quite surprising to note that the hippocampus only projects to two cortical areas and in very discrete fields. One is the retrosplenial cortex here, the other is the entorhinal cortex. So it's interesting that there are only two out channels from there, but then through intercortical interactions, memory traces become more and more widely distributed. So, try again? I'll try. Yes. So, my primary interest is actually in understanding how come that memories of stressful experiences come to cause negative affective states. As a field, as basic scientists, we have worked a lot on various animal models of anxiety and depression by putting animals in different situations and measuring how active or inactive they are. Our approach was to look at affective states through studying memory circuits. And the idea behind that was that it's not about the stress what makes the problem. It's about how we interpret, process the stressful situation, how we encode it in memory circuits, and how then this activation of memory circuits engages the whole limbic and affective system. And of course, our main interest is to understanding the link between the two and why some memories of the same event in some individuals will cause severe distress and in others will go on unnoticed. But following this structure, we will kind of get slowly also to some aspects of pain and memory. And I think that nowadays the cognitive affective coupling is reasonably accepted by the field. However, it was not for many years an intuitive thing because, as you know, probably it was always the rational brain and the irrational brain. And I do believe that the field now accepts that even what sounds most irrational, it's still rooted in cognitive processes or in the way how we interpret the world around us. So we are interested in the fundamental mechanisms of memory, how they contribute to adaptive and maladaptive behavior. And really our end goal is to identify treatments based on the mechanisms that we hope to discover. Now, when it comes to memory systems, they are defined in many different ways. And this is really the oldest, most basic classification, which is actually based on cognitive analysis in human subjects. And although the boundaries are really not as sharp as we always present when we classify things, these classifications still more or less hold. So we have memories which we can express, that we can talk about. These are memories of our experiences, or which are episodic memories, or memories of facts, when we know the birth dates of certain people, events that happened which haven't affected us, facts about which city is the capital of which country, and so on. So that's all in the declarative memory system. And all of these memory circuits are conscious memory circuits. So we actually can only process these memories if we have reasonably functional and coherent state of consciousness. And then there is also a vast memory system, which is primarily subcortical, which includes non-declarative memories. These are memories, for example, of simple conditioning events. Sometimes, for example, if you go to the dentist and you never even think of that smell, but that smell becomes ingrained. And whether or not you're conscious about it, it can trigger some negative reaction later on. Or skills, all the skills that we acquire. Although all of this is really not so strict in a way that we do need to have conscious awareness when we start learning. For example, motor skills. If you learn a bicycle, of course, in the beginning, you are struggling and trying, and there is certainly intent to that. But once the skills are acquired, we perform them without any need for conscious awareness or any intentional effort. And so these are just these various types of learning which do not rule out consciousness, but they can be performed without it. Yes.
How do we really know the role of consciousness in these things?
What we know is that, for example, in at least in patients who cannot perform hippocampal or cortical tasks, they have completely intact procedural memories, like in Alzheimer's disease, for example. This is why this classification comes from Larry Squire, who worked with a lot of people who either had deficit in one domain or another. And he would see that in those who are completely deficient with declarative memory have intact non-declarative memory, and in some rare instances, the other way around.
So those patients, they're still conscious? Or...
Yes. They're still conscious, but they have big chunks of the brain, which are not of the cortex, which are not able to provide access to the memories.
So then where does the consciousness bit come in?
The consciousness gets into the ability to intentionally access the memories. So that intent is disrupted. I mean, it's not that they are unconscious, but they cannot perform operations which typically require intact cortical conscious systems. It's not that they're unconscious, it's just there are many operations of consciousness which are self-awareness and awareness are only some of them. Being awake is only one of them. So there are many, many operations. One of them is intentional search through the memory circuits. Not really planning, it's really getting access. So they can't access the memories which are distributed around the cortical networks. That's what it meant in this case.
Okay. So it's not consciousness per se, it's...
No. It's certain aspects of conscious operations. Yes, sorry that I took it too liberally. So it's conscious operations. Now, that was what we know about memories at the cognitive level. But if we get down to the neurobiology, what are actually memories? What do we think when we talk about memories as a readout of brain function? And the best we can say so far is that memories are patterns of activity generated by discrete neuronal assemblies, like those schematized here. And in the episodic memory circuits, such patterns not only help us to reconstruct and remember and, in a way, remake past events, but also to re-experience them. That's a very important component of episodic memory systems. It's not only remembering, it is also re-experiencing. And this will become important when we talk about pain. Of course, I don't know how much the pain field has dealt with this, but this has been a very important topic in episodic memory research, especially when it comes to trauma and traumatic memories, where subjects, for example, suffering from PTSD, keep on re-experiencing the instances of their original trauma. Now, when we form a representation of an event, we probably encode many different things, but this is really a very simplified summary of what I believe are the three essential components that take of memory formation and memory representation. One is that we remember the features of the world. We represent the world itself as it is, without any evaluation, whether it's good or bad, and so on. So we will remember the configuration of this room, which chair is in the corner, which is there, the object locations, relationship between these things. So basically, it's somehow a representation of our physical space. The second thing that we remember is its value. And this is where our brain kicks in really in now different way to modify the representation of the world. We give it an assignment. We make it to be positive or negative, or sometimes we don't, it is simply neutral. Now, because I'm a basic scientist working with mice, we always have this component because it is very difficult to study neutral memories in animals. So we always have to throw in some reward or punishment in order to have them behave in a certain way. And through their behavior, we measure what they remember or not remember. So, but that's what we do as humans, actually, all the time. We encode a situation, and we encode its value. And that value depends on how we evaluate that situation at the moment, but also based on our prior experiences in similar situations especially. So our past history can very much influence how different individuals encode the same event. This is why we have so much variability. And then the third component is the internal state. And although I will only touch on this, I will really mention, internal states are really very important components of memory representations. For example, if we are sad, we are more likely to encode events as negative, and vice versa if we are happy. If, I don't know how many of you have been on corticosteroid hormones for allergies and so on, they completely change the states, people, or on alcohol. People sometimes, when they're drunk and forget their keys, cannot find them until they're drunk again. So you have this state dependence, which significantly determines our access to the situations. Yes, it's a pity. There were a lot of studies on that in the 70s and 80s, and somehow it stopped. But and these three different components of memories enable us actually to do three very different things. The first one helps us differentiate between these different stimuli. That's how we know this is whole A and not whole 2 or B or C and so on. The value is critical in organizing our behavior in that situation. If we like it, we will approach, we will like to be there, we'll feel good there, we'll want to be there more and more. If we don't, we will try to avoid it as much as possible. internal states play a key role in accessibility to retrieval. So as I mentioned, one was the finding the key, but in many different ways. You probably have noticed that sometimes you can remember things easily, some other times you don't, but then after that remember. Typically, if you are in a state which is similar to the original state, the access to memories is the easiest. So this is not exclusive. Sometimes sometimes there is complete amnestic barrier and you can't remember anything unless you're in the same state. Sometimes it's a gray zone, so remembering is easier or more difficult. So now that we've said that we need to have these specific patterns, the question is, how do we generate these patterns? And for a long time, we all thought that we have to instruct the nervous system how to remember something, so as if we, through plasticity, have to learn new things. And it turned out that this doesn't have to be the case. And this is not an accepted view of the field. This is my view of the field because I was trained as immunologist. So my road to neuroscience was very gradual. And as you know, in the immune system, we had a very similar theoretical construct that we thought that we teach the immune system to recognize foreign antigens, until it was discovered that in the immune system we already have immune cells which can recognize any possible antigen out there. And I think that we have exactly the same thing in the brain. It started, this view is motivated by the work primarily of Gerald Chan, who, when he started working on genetic diversity of neurons, everybody thought that this was artifact and nonsense. And today, now 18 years later, we have a consortium of 18 biggest universities in the United States who got billions of dollars to study this. How different is one neuron from another? In mice, the idea is that they have about 100 genes difference. In humans, 1,000. Now, if all of these neurons are different, this means that we already have every possible neuron that can respond better or worse to the stimuli that get in the brain. And obviously, those which are best tuned to the stimulus are more likely to respond than those that are not. So we already have huge diversity in the brain as it is, which in some crazy way means that we already have all information we need. So what is it that we are actually learning? We are learning to make sense of this information and to put the things together in meaningful blocks, which we will then revoke and reconstruct as we need during life. How do we do that? The neurons and the brain in general, the nervous tissue, has many, many ways to put these puzzles together and these little building blocks. One is by changing their signaling. So just look at one single neuron that learns. You will see that in the beginning they come with certain signaling pathways, then the next time they will not. Once they learn, they don't have to do that. Then if the things change, they will change the signaling, which I will show in a moment. Another way is through the buildup of perineuronal nets. This I will talk about a little bit more later, but perineuronal nets are really an interesting way how to organize cells into assemblies. Because they're around interneurons, they will stabilize the innervation field of an interneuron. And as you know, once you have a stable interneuron field, then the same cells will either be excited or inhibited. So you have a very easy way to generate a pattern which can be repeatedly evoked. The most famous way how we can group neurons into assemblies is through long-term potentiation or depression, which is known as synaptic plasticity. And then we have through coordination of neuronal spiking with waves generated by interneurons. And lastly, by using the same brain waves to coordinate the activity of different brain regions. So as you can see, there are so many levels by which we can organize information to generate specific patterns. And we rely on all these mechanisms in different ways, in different situations. So what do? In my lab, we don't do spared nerve injury, but we do something which at first sight doesn't look similar at all. And I hope I'll convince you that actually it's not that different at the end. So we do fear conditioning. And we do context fear conditioning, which is we put the mice in a box, which we call environmental context. We call it a context to differentiate it from a cue, like a sound or a flashing light and so on, because it is more complicated to create a whole representation of environment, and it depends on the hippocampus. It critically depends on the hippocampus, which is not the case with simple cues. And if at the end we deliver a shock, these animals will acquire freezing behavior, which we can see if we put them back in that box the next day. Now, what is really important to know about this freezing behavior is, first of all, it's specific. It will happen here, but not if we put them in a different box. So this is already one important feature of well-encoded memory. Second, it's associative. They need to start, this shock has to come at the proper time when the memory of the context is started to form. If we just deliver the shock in the beginning, they will not learn anything. Yeah, if you see most graphs that I'll show will have this freezing behavior here, and then this manipulation will be up or down or unchanged depending on what we are measuring. So this graph shows how they freeze in the original box here, but not in the box here. This graph shows how when we deliver the shock immediately, they will not learn. So it's highly associative. Second, it's persistent. If we deliver this shock once and look at the mice 6 months later, they will still freeze, which is an incredibly long time for a rodent. On the other hand, for us, it's really a unique opportunity. We don't have many types of learning in rodents where we can look at memories over such a long time. And lastly, although this fear response can persist, we can actually change its trajectory. So if we put the mice back in a box again and again and again without the shock, with the time, their freezing response will go down. For a while, scientists have thought that this could be a forgetting and weakening of the original memory. However, we don't think that this is the case. We think that the mouse end up having two memories. One, that there is a shock, another, there is no shock. And then we can actually make them recall one over the other to show which of them is dominant. For example, if the mice extinguish fear, as we can see here, the freezing response goes down, a very brief reminder of the shock will bring it up. And I'm explaining this in more detail because I will talk about this paradigm a little bit in a moment. Lastly, well, this could be a bit of an overkill, but I'll say it anyway. We can do one last thing here, well, probably more, but one which is important for today, which is this. If we, as I mentioned to you, if we give the immediate shock, the animals will not learn. However, if we put the mice in the box the day before this shock, they will learn. And this has been a phenomenon that has puzzled the field for a long time, until a series of studies demonstrated that what happens here is that mice first learn the context and then actually can get conditioned to the memory that they've acquired the day before. Which is really interesting. It's that they are now not forming one negative memory, but they are associating a negative value to a memory which was formed the day before. This paradigm is really important because we can manipulate this phase or this phase independently, and then we know whether the learning affects the encoding of the context or the assignment of the value. So if we want to tease apart these different components that I mentioned about, this paradigm is very useful. So to summarize and put this all back in the story of the paradigm, we can see that through the specificity features, the mice can remember this box where they got the shock from boxes where they don't get the shock. They organize their behavior. So why do our mice freeze? They freeze because they can't do anything else. If we put a little opening in this box, they'll run away. They will not freeze. So you can see that they don't acquire a reflex or one conserved response. They really adapt to they learn and they express a whole behavioral repertoire depending on the options they have at the time of the test. And lastly, we know that the change that they can recall these different things and reinstate the state of fear if they just get brief reminders, which can put them back at the state of encoding. So even after extinction, if we just put a little shock reminder, they can go back to the internal state and then completely, fully recover the old memory. And using this very simple paradigm, we can go from behavior, which I mentioned, which is believed to be highly adaptive, to situations which can be entirely maladaptive and reminiscent of pathological phenotypes in humans. For example, if they start freezing to different boxes, this is known as generalization. And this is found very frequently in various patients with anxiety disorders, as well as in patients with depression. They can develop resistance to fear extinction. So we have a genetic mouse line where we can put the mice up to a month without a shock, and they will still freeze as if it was the day before. That is very common to patients with PTSD. We can induce dissociative amnesia using various drugs where we can control their access to the memory systems, and so on and so forth. So these are the different models that we study. Now, what we have looked at, is it already 11? And I'm done?
Well, yeah.
Then I can go to the conclusion right away. This was too long. I'm so sorry. Anyway.
Okay. Just if you can squeeze it.
I will squeeze it very much. I'll have to squeeze it. So we have looked at all kinds of genetical and molecular mechanisms. But what is important to say that although the hippocampus is a key to the beginning of the memory encoding process, what happens later all becomes cortical. And with the time, the hippocampus disengages. And now I'll go to the experiment that I wanted to highlight, which is what I did with Vanya and with our colleague Jack Kessler, who is working on hippocampal neurogenesis. So these are 4 groups of mice. This is a control. And these are newly generated neurons in the dentate gyrus. And these are astrocytes in the red GFAP. So these are normal animals. These are also wild-type animals that are running. They are running over a month. And you can see they have increased neurogenesis, so the green signals are really strong. They're also very strong if we overexpress a protein called noggin, but they completely disappear if we overexpress a protein called BMP4. So we can change the state of the hippocampus by changing the level of neurogenesis. What happens there is that the animals which have increased neurogenesis show better learning in every paradigm that we've looked at. Object recognition, spontaneous alternation, novel location recognition, contextual learning. So they're really very good. And it's exactly the opposite with those which are the blue ones, which are overexpressing BMP4. So they're impaired in everything. And for us, this was really a very good model to see, there is another thing. So they can't form memories. However, they are highly depressed. In every test of depression and anxiety, they are actually performing really on the more depressed side. So they are immobile, they are not motivated to cope, they are not motivated to do anything. And we thought this would be a really good thing to see whether transition from acute to chronic pain depends on learning or it depends on their affective state. And I think most of the people involved were betting on the affective state. They thought that these animals will have more pain because they're more depressed. And what we saw was exactly the opposite. And I think this was one of the strongest modification of learning that we've observed in this model. These are animals who transition from acute to chronic pain. You can see in 14 days, already by day 4, they have a very low pain threshold. If we remove, these are the BMP one, look at the red ones, which are the ones that can't learn. No, this must be, wait a minute. Sorry, this is the noggin. This is mislabeled. No, apologies. It's been a long time ago now. These are the ones that can't learn, but they're depressed. These animals don't transition to chronic pain at all. So although they show every sign of a mouse depression, but are not capable of learning, they are not capable of transitioning to chronic pain. And not only in the spared nerve injury model, we had exactly the same with the Karagrinan model, which is inflammatory. And they do start, they have normal intact acute pain response. However, they recover very quickly, and after that, they're fine. And together, which I don't have time for this, unfortunately, I'll just come back to the conclusion. Together with all the stuff I didn't show, what we believe is shared by pain and by negative affective states such as fear, which are rooted in learning, is that they are both based on initial negative event. They persist far beyond this initiating event. The representation in brain circuit increases with time, and their reactivation can only increase their representation in the brain. And they depend on really, at least in the first phase, when they're encoded, much more on hippocampal memory systems than on negative affective states. This later on will change. We know that once they stabilize in chronic pain, then the hippocampus may or may not be as involved. We are not sure yet. But in this transition, we clearly know that it plays an important role. And Johan actually, from our side, was the person who did most of the experiments, but everybody was involved. And thanks to Vanya here especially, because without him, I would not have done this. Thank you.